Hi, everyone, and welcome to this webinar. My name is Jolie, and I will be presenting Introduction to Illumina Sequencing by Synthesis. This is a great high-level overview of sequencing by synthesis technology for anyone who is new to the technology or who would just like a refresher. So let's get started. I'll get started by introducing the 4 basic steps of the Illumina sequencing by synthesis workflow. First is library preparation, where adapters are added to the ends of the DNA fragments to be sequenced. Next is cluster generation, where the library is hybridized to the flow cell and amplified to make many copies of the DNA fragment. Then sequencing is where the instrument incorporates nucleotides into the library sequence and records the growing chain of DNA sequence. Finally, data analysis is where base calls are made and aligned to the reference genome. I'll go into details for each of the 4 steps of the sequencing workflow throughout the webinar. Let's first look at library preparation. The purpose of preparing the library is to make the extracted DNA compatible with binding to the flow cell for sequencing. Generating a library can be done using many different kits from Illumina, and each kit will produce a library that is compatible with Illumina sequencers. Check out the Illumina library prep kits and associated index kits for Illumina kit options using the link below. First, we start with large double-stranded DNA that has been extracted from the sample being investigated. The DNA is then fragmented either enzymatically or physically to produce fragmented DNA. The fragmented DNA then has 2 unique adapters added to the ends, resulting in a complete library. Based on the amount of DNA input, this prepared library pool can then be amplified using PCR to increase the number of libraries. This forms a library that will bind to complementary DNA fragments or oligos on the flow cell lawn. Let's take a look at the full library structure. Each complete library will have the DNA insert, which is the fragment of double-stranded DNA from the original sample, as well as the adapters on each side. The 2 unique P5 and P7 adapters are on both ends of the DNA insert and contain the binding regions for clustering, the primer binding sites for sequencing, and the indexes for multiplexing. The binding regions for clustering include the P5 and P7, which are complementary to the flow cell oligo lawn, and these facilitate library hybridization to the flow cell. The binding sites for sequencing include the read1 sequencing primer binding site for the forward read and the read2 sequencing primer binding site for the reverse read. A single read library would only be read in the forward direction, and a paired end library would be read in both the forward and reverse directions to increase coverage. Lastly, the indexes allow for multiplexing, which is the ability to combine multiple samples in a single sequencing run. Indexes are a short sequence of DNA, normally about 6 to 10 base pairs, that barcode or uniquely identify samples within a run. In this example, 2 indexes are present, signifying a dual index library. If there were only 1 index, the library would be a single index library. Please see the Illumina adapter portfolio link at the bottom of the slide for more details on Illumina library adapters. After the libraries are prepared, the next step in the Illumina sequencing workflow is cluster generation, where the prepared libraries are amplified on the flow cell and form a cluster that will be imaged during sequencing. Let's first define what a cluster is. A cluster is a group of about 1,000 clonal copies of the same DNA library bound very closely together in the flow cell. The multiple copies in close proximity are required to produce sufficient intensity and signal to be recognized by the optical systems. Each cluster begins as a single DNA library and is then amplified to produce the group of clonal copies. During the sequencing run, all strands within the clusters are imaged during each cycle. Here's an example thumbnail image from a sequencing run. Each white dot is a cluster producing signal, and the black regions represent the areas of the flow cell where clusters are not present. The instrument uses images like these to determine the location and intensity of clusters and call what bases are incorporated. Let's also define a flow cell and the purpose of a flow cell during sequencing. The flow cell is the surface where cluster generation occurs and is essentially a sandwich of two glass slides with channels to form lanes. The inside of the lanes are covered in short DNA sequences known as oligonucleotides. These flow cell oligos are complementary to the P5 and P7 sequences at either end of the library and allow the library to bind to the flow cell. When libraries are flowed through each lane, they bind to the oligos. This image in the top right shows a high-seq flow cell with 8 individual lanes, but the concept is the same for flow cells on all systems. There are two types of flow cells, non-pattern and pattern. Non-pattern flow cells are used on the MiSeq, MiniSeq, NextSeq 500, 550, and HiSeq 2500. And pattern flow cells are used on the iSeq 100, NextSeq 1000, 2000, HiSeq 3000, 4000X, and NovaSeq 6000. On a non-pattern flow cell, the surface is flat and oligos randomly cover the flow cell such that libraries can cluster anywhere. On a pattern flow cell, the surface contains billions of nanowells at fixed locations, and oligos are limited to the wells so that single libraries can only cluster within the wells. One advantage of pattern flow cells is that clusters are contained in the nanowells, which allows for an increased cluster density, resulting in greater overall output. Here we have an example HiSeq 2500 flow cell on the left and a HiSeq X flow cell on the right. The only visible difference when looking at them by eye is the iridescence on the surface of the pattern flow cell. Within these magnifications, you can compare the non-pattern flow cell with random clusters and the pattern flow cell with neatly organized wells where single libraries will bind. Another example is the non-pattern NextSeq 500, 550 flow cell on the left and the pattern NextSeq 1000, 2000 flow cell on the right, which also differ in shape and size. Now let's look at the process of cluster generation. Cluster generation occurs on board the instrument for all instruments except with some of the legacy HiSeq systems. Let's first look at traditional clustering on non-pattern flow cells and then go over cluster generation on the pattern flow cells. First, the library fragments are denatured to form single-stranded libraries. On non-pattern flow cells, these libraries attach randomly to the complementary oligos inside the flow cell lane. So if there are two strands of DNA in close proximity, clusters will form nearby and could even overlap, developing a cluster with more than one DNA strand. Additional clustering regions are delivered to the flow cell and the libraries are primed from the flow cell oligos. A polymerase extends a complementary strand of the hybridized fragment through the full length of the library. Priming in this sense means that the flow cell oligo is the starting point for building the cluster. After extension from the flow cell oligo, the library is double-stranded and includes both the original template as well as the newly synthesized strand. This newly synthesized strand is covalently bound to the flow cell via the oligo, and the first synthesis of the new strand of DNA is now complete. Then the double-stranded DNA template is denatured and the original template is washed away, leaving only the newly synthesized single-strand DNA anchored to the flow cell. This single DNA strand must be amplified so that the fluorescent signal reaches a detectable level of intensity during sequencing, and bridge amplification is the process that generates the clonal clusters on the flow cell. Bridge amplification begins when the DNA molecule bends over and attaches to a neighboring complementary oligo on the flow cell lawn. Then the DNA strand is primed off the oligo and another strand is synthesized by extension while in the bridge position. After the extension is complete, there is a fully formed double-stranded bridge. This double-stranded bridge is then denatured so that there are two single-stranded templates covalently attached to the oligo lawn. This process of bridge amplification doubles the number of libraries on the flow cell and generates a forward and reverse strand. The process is then repeated. During the next cycle of bridge amplification, these two single strands of DNA bend over and are once again extended. This results in 4 single strands of DNA template that are covalently bound to the flow cell, with half the strands in the forward direction and the other half in the reverse direction. This process is done multiple times until there are thousands of double-stranded bridges on the flow cell surface. And this process is how a single strand of DNA turns into a single cluster on the flow cell with many clonal copies of DNA. Once the amplification is complete, the bridges are denatured, resulting in only single-stranded DNA anchored to the flow cell. The libraries are currently attached to both the P5 and P7 oligos on the flow cell lawn, which means that the strands are in both the forward and reverse complement directions. When sequenced, the DNA libraries will be primed at the top of the strand and then will be read toward the flow cell. If the libraries are attached to both the P5 and P7 oligos, the DNA will be read in both the forward and reverse direction. Let's use my name, Jolie, as an example. In the forward direction, we have sequences that would be read correctly, J-O-L-I-E. But we also have libraries in the reverse direction, E-I-L-O-J. In this case, the instrument would not be able to determine what is being sequenced because both J and E are being called during the first cycle. Therefore, for sequencing, we need all the strands within the cluster to be read in the same direction. To allow the instrument to call the correct base and read in one direction, the libraries in the reverse direction are cleaved, and only the libraries in the forward direction remain. To prevent DNA template strands from bridging again or further extension from the 3' ends of the flow cell oligo, the 3' ends are blocked. Now that only forward strands remain on the flow cell, read 1 is ready to be sequenced. To start read 1, a sequencing primer is hybridized to the library, and the DNA template is built from this sequencing primer in the direction toward the oligo lawn. Pattern flow cells cluster slightly differently than non-pattern flow cells because they use exclusion amplification, or XAMP technology, within a nanowell, but the concept of amplifying a single strand of DNA to form a clonal cluster is the same. Let's take a closer look. The pattern flow cell is covered in nanowells, and these nanowells help ensure even cluster size and spacing. They also allow clusters to be more tightly packed on a flow cell, increasing the output in a smaller footprint. Complementary oligos are present only inside the nanowells and are not found outside the nanowells. As with non-pattern flow cells, single-stranded DNA template is flowed across the flow cell, and one single-stranded DNA hybridizes to the complementary adapter. Polymerases immediately extend the template, creating double-stranded DNA. Double-stranded DNA is denatured and concurrently hybridized to the flow cell. After double-stranded DNA is formed, the process of denaturing and rebridging is repeated. This denaturing and bridging repeats until every oligo within the nanowell has a DNA template covalently attached. The XAMP technology utilizes nearly instantaneous amplification to ensure that only a single DNA template binds and forms a cluster within a single well. Next, to prepare for sequencing, the DNA strands are linearized, and the strands are still in both the forward and reverse directions. The reverse strands are then cleaved, leaving only the forward strands. The Re1 sequencing primer then hybridizes to the forward strand, and the DNA is ready for sequencing. Now that cluster generation is complete, sequencing can begin. During sequencing, individual bases are incorporated as the growing strand is imaged. All Illumina sequencing platforms utilize the sequencing by synthesis, or SBS, workflow, but 3 different detection methods have evolved: 4-channel, 2-channel, 1-channel chemistry. Let's go over each method in detail. 4-channel chemistry is used on the MySeq and all HiSeq systems. In 4-channel chemistry, each of the 4 bases has its own fluorescent dye and produces unique signal when excited by a light source, as shown here. During each cycle, bases are incorporated. Then the instrument pauses, excites the libraries with a light source, and captures 4 distinct images using 4 different wavelengths, corresponding to each base. All G's, T's, A's, and C's will be seen in a single picture for that cycle, and the strength of signal within a given wavelength is what determines the base call. Let's walk through this diagram showing SBS incorporation in 4-channel chemistry. This blue strand on the left is the DNA template, and this small purple sequence is the read primer. In the first cycle of chemistry, fluid containing all 4 fluorescently tagged nucleotides are flowed over the flow cell. Here you see red for thymine, blue for adenine, yellow for cytosine, and green for guanine. The matching base is incorporated to the complementary strand. All fluorescently labeled nucleotides include a block that prevent addition of more than one base during each cycle. After the new base is incorporated, the unused nucleotides are washed away. The instrument pauses, and a light source excites the newly added base, and a series of images in each wavelength is taken of all clusters. After these images are taken, the base is deblocked and the fluorescent component is removed. By deblocking and removing the fluorophore, the next base can be incorporated into the growing strand. The process of incorporate, excite, image, and cleave is repeated until the number of cycles for the read is met. For example, for a read length of 100 cycles, this process will repeat 100 times. As sequencing occurs, a new strand of DNA is synthesized, which is why this process is called sequencing by synthesis. The next type of chemistry is called 2-channel chemistry, which is used on the MiniSeq, all models of the NextSeq, and the NovaSeq 6000. Instead of using a separate dye for each base, Two-channel chemistry uses two dyes and a combination of images from two wavelengths to detect nucleotides. For the traditional red-green chemistries, thymine will appear only in the green channel, cytosine will appear only in the red channel, adenine will appear in both green and red channels, and guanine does not produce any signal and will not appear in either channel. The concept is the same for the NextSeq 1000, 2000, but the green and blue channels are used, which improve resolution. An intensity plot will be generated with this information, and the incorporated base is determined by the corresponding position on the intensity plot. The intensity plot for the NextSeq 500, 550 uses the red and green axes, and the intensity plot for the NextSeq 1000, 2000 uses the green and blue axes. Since only two images are taken, the amount of data processing and runtime for sequencing is reduced. Now let's review the diagram showing incorporation in two-channel chemistry. In the first cycle of chemistry, all four bases are flowed over the flow cell. The complementary base is incorporated one base at a time. After incorporation, the unused nucleotides are washed away. A light source excites the incorporated bases, and in two-channel chemistry, only two images are taken in each wavelength. After imaging, additional reagents are flowed over the flow cell to deblock the base and knock off the fluorophore, and this process is repeated until the read is complete. Based on the intensity plot from either the red and green channels or the green and blue channels, we can determine which base was incorporated. As with four-channel chemistry, the process of incorporate, excite, image, and cleave is repeated for the number of base pairs in the read. Lastly, we have one-channel chemistry, which is used on the iSeq 100. In one-channel chemistry, two images are taken in a single wavelength, but bases can be distinguished from each other because of an additional chemistry step. This chemistry step results in the presence or absence of signal based on the incorporated nucleotide. The signal is either on or off in each image. Once the first image is taken, a chemistry step will either cause no change to the signal being produced by the fluorophore, or will change the signal by knocking off or adding a fluorophore. Based on the signal found in both images, the instrument can determine which base was incorporated in each cluster. For instance, for thymine, there is signal in both images and no change during the chemistry step. For adenine, there is no fluorophore during the first image that is knocked off during the chemistry step, resulting in no signal in the second image. For cytosine, there is no signal in the first image, and the chemistry step adds a fluorophore, resulting in signal in the second image. Guanine produces no signal in either image because there is no fluorophore, and there is no change in the chemistry step. For more information on one-channel chemistry, please see the technical bulletin in the link. Let's take a look at SBS incorporation with one-channel chemistry. Similar to four- and two-channel chemistry, at the beginning of the cycle, reagents containing all four bases are flowed over the flow cell, and the complementary base is incorporated to the template strand. After incorporation, the unused nucleotides are washed away. A light source excites the incorporated bases, and the first image is taken. This is where one-channel chemistry differs from four- and two-channel chemistry. More reagents are flowed over the flow cell for the additional chemistry step, resulting in knocking off a fluorophore, adding a fluorophore, or not changing the signal. Another image is then taken, and based on the differences in signal between the two images, the instrument can determine which base was incorporated. In one-channel chemistry, there is the same workflow of incorporate, excite, image, and cleave, but there is the additional chemistry and image step before cleaving. To recap, here's an overview of the sequencing chemistries. In 4-channel chemistry, each base provides unique signal from one of 4 fluorescent dyes. 4 images are taken, and there is only 1 chemistry step per cycle. Bases are called by the unique wavelength for each nucleotide. In 2-channel chemistry, 2 images are taken in 2 wavelengths, and there is only 1 chemistry step per cycle. Bases are called by the combination of signal in the 2 channels. In 1-channel chemistry, 2 images are taken in a single channel, and there is a 2nd chemistry step at each cycle. Bases are called by the varying signal in each image caused by the chemistry step. Now that we understand the different types of chemistry, let's discuss paired-end sequencing, which allows you to increase the amount of data recovered from your sequencing experiments by reading in both directions. We've covered cluster generation and how clusters are sequenced in each cycle. After all the cycles are sequenced in read 1, the clusters will undergo paired-end turnaround to sequence read 2 in the reverse direction. Before paired-end turnaround, the original template DNA is attached to the oligo, and the read 1 primer and read 1 sequence strand are hybridized to the template DNA. To prepare for paired-end turnaround, we need to wash away the read 1 primer and read 1 sequence strand, and then deblock the 3' end to allow the DNA strand to form a bridge with the oligos on the lawn. Similar to bridge amplification, the template strand first bends over, then it is primed off the oligo and is extended to the full length of the library. This results in double-stranded bridge DNA with both forward and reverse directions anchored to the oligo lawn. The double-stranded DNA is then denatured to allow the strands to become linearized, single-stranded reverse complements that are both attached to the flow cell. If we bring back the example with my name, read 1 has already sequenced the forward direction of my name. Now we need to cleave off the forward reads to allow the instrument to sequence the data in the reverse direction in read 2. After the forward strand is cleaved and washed away, only the reverse strands remain. The 3' are blocked once again to prevent bridging. The read 2 sequencing primer is hybridized to the adapter strands, and sequencing begins in the reverse direction. Earlier in the webinar, I mentioned multiplexing and how it can be used to combine multiple samples into one run. Let's now go over how indexes are used for multiplexing because there are a variety of methods. The first and simplest indexing strategy is called single index reads, and this method can be performed on all platforms. An index is a short sequence of DNA that is included within the adapter and is unique to each sample. For the single index read method, there's a minimum of 2 sequencing reads, read 1 and index 1. And there could potentially be 3 reads if a reverse read has been included in the run setup. For all the methods presented here, let's assume there is a read 2, but read 2 is not required. This protocol shows read 1, index 1, and read 2. Let's walk through each read here. The read 1 primer is first hybridized to the template strand, and read 1 is sequenced. The read 1 primer and sequence are then washed away. The index 1 primer is hybridized while the strand is still in the forward direction, and index 1 is sequenced. The index 1 primer and sequence are then washed away, and then paired-end turnaround occurs. After paired-end turnaround, the forward strand is washed away, and the read 2 primer is hybridized and extended to sequence read 2. The next indexing strategy is dual index reads using the forward strand method, which is used on the MiSeq, HiSeq 2500, and NovaSeq 6000 with control software version 1.6 or earlier. Dual indexing allows for more samples to be multiplexed because 2 indexes can be used to create many unique combinations, which are then used to identify each sample within a cluster. If read 2 is sequenced, there are 4 reads during a dual index run: read 1, index 1, index 2, and read 2. In this forward strand workflow, index 2 is primed off the oligo lawn and is read in the forward direction, since sequencing occurs before paired end turnaround. Here, read 1 is sequenced and washed away. While the strand is still in the forward direction, index 1 primer is hybridized in sequence. The index 1 product is then washed away. The DNA strand bends over and index 2 is primed off the oligo lawn because the oligo acts as a primer. Index 2 is read and the strand is completely extended to produce the reverse strand. Paired end turnaround is then completed after index 2 is read. The read 2 primer hybridizes to the flow cell and read 2 is sequenced. The iSeq 100, MiniSeq, NextSeq systems, HiSeq 3000, 4000X, and NovaSeq 6000 with control software version 1.7 or greater use the dual index reverse complement workflow. This workflow uses 2 unique indexes to identify samples and has 4 reads per sequencing run when read 2 is sequenced: read 1, index 2, and read 2. The difference between the reverse complement workflow and the prior forward strand workflow is that here, index 2 is primed using an index 2 primer, whereas previously in the forward strand workflow, index 2 was primed off the oligo lawn. Index 2 is then read in the reverse direction since sequencing occurs after paired end turnaround. Let's break it down. Here, read 1 is sequenced and washed away. Index 1 is sequenced and washed away. Prior to sequencing index 2, the DNA undergoes paired end turnaround so that the DNA sequence is now in the reverse direction. Once paired end turnaround is complete, the forward strand is washed away. An index 2 primer is hybridized to the library, and index 2 is sequenced in the reverse direction and is then washed away. Read 2 is then sequenced. For more information on the single index and dual index reads, please reference the Index Sequencing Overview Guide. Now let's look at the final part of the sequencing workflow, which is data analysis. Now that sequencing has completed, primary analysis analyzes the signal from the images on the control software. First, a template map that identifies the location of every cluster in the flow cell is generated. Once the location is determined, the instrument control software will process the raw TIFF images into CIF files. The instrument control software then extracts the signal intensity from the CIF files and converts them into base calls for each cluster in the BCL files. The instrument control software also determines the quality Q-score for that base call, which allows us to determine how confident the instrument is in that base call. Let's briefly cover what a Q-score is. The Q-score is a quality metric on the probability that the base call made for a cluster is incorrect. During a sequencing run, the Q-score will only be calculated for the clusters that pass the quality filter, which is determined after the first 25 cycles. Illumina uses a metric called Q30 to determine the quality of a sequencing run, which is a measured average of all base calls throughout the run. And a base call with a Q-score greater than 30 has an accuracy of 99.9%. Illumina has produced Q30 specifications for each instrument based on the run length of the sequencing run. For example, a MySeq 2x300 run has a specification of greater than 70% Q30. Run specifications can be found on the support pages for each instrument, which is linked here. For more information on Q scores, see the links at the bottom of the page for the documents Quality Scores for Next-Gen Sequencing and Understanding Illumina Quality Scores. Let's also briefly address what demultiplexing is. During sequencing, there are millions of reads representing all the fragments from many pooled libraries. Demultiplexing is post-run analysis that separates and sorts those pooled library fragments based on their index sequences. And this is what allows the index sequence of many samples to be determined. After a primary analysis is complete, the base calls, or BCL files, are then demultiplexed and converted to FASTQ files via the onboard secondary analysis software, either using the BCL to FASTQ command or within BaseBase. FASTQ files can then be fed into tertiary analysis programs to align the reference genome, perform variant calling, or perform de novo assembly, which help generate conclusions from sequencing experiments. Before I finish the webinar, I'd like to introduce some great resources that will help you plan your experiments. Based on the application you are planning to run, there are many different library prep kits to choose from. To assist, Illumina has created a helpful tool called the Illumina Library Prep and Array Kit Selector to help narrow down the best kits for your project. When using the kit selector, you can select from area, application, method, species, and system. For instance, I can input that my area of work is drug discovery and development, my application is gene expression, my method is mRNA, the species I'm working with is rat, and I have the MySeq and HiSeq 2500 available to use. Note that you can put multiple options in each category, and here I've selected both MySeq and HiSeq 2500. I could have also chosen more selections from the other fields. I'm then given two kit options, TrueSeq RNA Library Prep Kit version 2 and TrueSeq Stranded mRNA. Here I can compare these kits, and because of the tool, I'm able to quickly narrow down the best usable kits based on the requirements for my project. The link is provided here to the Library Prep and Array Kit Selector. To start sequencing, you will also need to choose which instrument you would like to sequence on. As a general guideline, the main difference between the instruments in Illumina's portfolio is the amount of data that will come off the sequencer. Our smallest sequencer, the iSeq 100, produces the least amount of data. For instance, a 2x150 cycle run will generate about 1.2 gigabases of data, while our highest output instrument, the NovaSeq 6000, will produce up to 6,000 gigabases for the same run length if running flow cells on both sides of the instrument. Here, the 5 instruments on top are the benchtop sequencers, which only run 1 flow cell per sequencing run. The 4 instruments on the bottom are the Illumina's production scale sequencers, which can run 2 flow cells per sequencing run. To choose the best sequencer for your project, you will need to understand the application you're running, how much data you will need for your analysis, and how many samples you're running. We have a very useful tool called the Sequencing Platform Comparison Tool that can assist you in choosing the best instrument for your sequencing run. To start, you'll choose research use only, molecular diagnostics, or forensic genomics. The molecular diagnostics application will utilize our FDA-cleared MySeq DX or NextSeq DX to perform diagnostic assays. The forensic genomics application will utilize the MySeq FGX, which was developed for the forensic investigation field. By selecting the research use only application, You will be asked your primary interest and technique. For this example, my interest was cancer genomics, and my technique was large whole genome sequencing. This will show a comparison of the best instruments for this application. Here you can review metrics such as the data output, number of samples per run, cost, and runtime to choose the best instrument for your project. See the link here for the sequencing platform selector tool. Here are some useful links, including the library prep and array kit selector and the sequencing platform selector that I just mentioned. There are also links to additional documents like the Illumina adapter sequences document that contains the sequences for the Illumina indexes and adapter trimming, as well as the index sequencing overview guide, which goes over in detail the indexing strategies I spoke about for single and dual index runs. There are also links to training and support videos such as the recorded webinars, where you can view previous Illumina webinars, and a 5-minute video summarizing sequencing by synthesis.